Okay, well, now we get to hear from Matt Nelsen, who is coming to us from one of the world's best museums, the Field Museum right here in Chicago. And he's going to tell us about some of the wonders of lichen. Please take it away.
All right, thanks so much for having me. I'm really excited to be here today. I want to chat with you about a little bit of work I'm doing, but I also want to give you a little bit of some background on an organismal group I'm guessing most of you are not familiar with. So I'm going to talk to you about lichens. These things, this kind of schmutz you've probably seen on tree branches before. I've kind of passed around a few pieces of bark or branches, and there's a hand lens here, a jeweler's loop that Amanda has that you can look at if you want to look at them up close. We can deal with that later too if you're interested. But my interests basically are kind of threefold. I'm really interested in symbiosis and interactions between different groups of organisms. Ecology and evolution kind of dovetails nicely with that. And then kind of late in my undergrad years, I got really into fungi. They just kind of, I took a course kind of as a joke. I couldn't believe there was an entire class on fungi and fell very, very hard for it and have continued to be interested in them. And so for me, the sweet spot is where all three of these really kind of overlap. And some of the questions I get especially excited about exploring are what roles have fungi played in shaping Earth over kind of geological timescales? How did some of these different symbioses evolve and what were the consequences of these interactions? So fungi, just for a refresher from some things you probably had in high school, but fungi are more closely related to animals than they are to plants. And like animals, they need to eat other things. And so the way they get their food is either by rotting, by decomposing dead things, or by forming kind of mutualistic or parasitic partnerships with living things. So I think they're a really cool model within which to try to understand the ecology and evolution of these symbioses. And then I also get excited about trying to understand how traits have evolved in different groups of these organisms and kind of shaped their evolutionary trajectories. So why are some groups more diverse than others? Those sorts of questions. I've worked in a range of systems, including ants and plants. You can kind of see on the bottom here a little bit with mycorrhizal fungi and wood rot fungi. But the type of interaction that really kind of first piqued my interest were these things called lichens, which are shown up at the top there. So there's this really long technical definition of what a lichen is, kind of boring and dry here. The crux of it is that it's a fungus and some algae. So this is one of these lichens. You can maybe see on that bark, you can see kind of a grayish version of that. But yeah, these algae tend to be small, little round green things that are pretty boring. And the fungus is not terribly exciting when it's on its own. So this is a fungus grown in the lab on a Petri plate. It kind of looks like Jabba the Hutt a little bit to me. And if compatible fungi and algae meet, the fungus kind of undergoes this transformation and makes this totally new structure now, has a totally different phenotype that will now house those algae inside of it. A simpler way to kind of understand this is that these are kind of like fungi that have discovered agriculture. So the idea being that the fungi are farmers, the algae are crops or livestock that they're raising. The algae are doing what algae do. They're photosynthesizing, making sugars, feeding themselves. And then they're also leaking some of that to the fungus. So the thought is that the fungus is making this house kind of to hold and kind of buffer those algae from the environment. And the algae in return are supplementing it with food. So this is kind of how this situation works. And this is something we all, accept without any question now, that it's, multiple different critters living together. But this wasn't always the case. So in the late 1860s, there's this guy named Schwendener who had a really crazy idea when he was looking through his microscope that all these little green things he was seeing inside this lichen was actually a different species. So prior to that, people had thought a lichen was one organism. And he said, no, I think it's two. I think there's a fungus and those green things are algae. And so he mentioned this, people got pissed. So essentially it was a lot of the lichen people got very upset. This is kind of a terrifying picture of Nylander, who is one of the foremost lichenologists of the time, he referred to this idea as the stupidity of Schwendener, and a lot of other people kind of went along with that as well. So Schwendener was poo-pooed, but eventually people came to accept that this, in fact, is, we are looking at multiple organisms. And so lichens have played a big role in our understanding of symbiosis. So when the term symbiosis was first used in these two papers here, lichens were cited as one of the examples of this new kind of system of these two different organisms living together. So while a lot of people might, think of them as this kind of weird, odd thing, they have been played an important role in our understanding of symbiosis since the very beginning. So as you're walking by the street trees and see these lichens, try to keep that in mind. And so these lichens have diversified into just a really wide array of shapes, colors, and sizes here. So we know of over 20,000 different species of fungi that form lichens. So for context, that's about as many species as there are birds and mammals combined. So people should care about the diversity of lichens. And you can just see they're really quite spectacular here. One of the things that's frustrating about them is that they're not real amenable to experimentation in many cases. So trying to culture these fungi is a very complicated affair, and trying to do all sorts of experiments on them can be really quite challenging, unfortunately. So some of the work that I'm going to be talking about today, an experimental approach would be ideal, but it's not really tractable.
20,000 fungus species. What about the bacteria?
Good question. So we know of about 200. We know there's a lot more out there, just like we know there's more fungi, but there's probably even, a lot more algae out there. Part of the problem is that they all look the same, or a lot of them do. They're kind of these little round green things. And so now with genetic data, we've been really able to kind of tease that apart. We're finding lots of hidden diversity we'd overlooked. But it's very much an asymmetric ratio where we have far more fungal species than algal species involved, which sets up some interesting questions about sharing, how we get very different fungi partnering with the same algal species and perhaps exhibiting selective pressures on that. So it's a really cool system. Before we go further, I know most of you are familiar with phylogenies, but I know we also have like physicists and all sorts of people from different backgrounds. So I'm going to be talking a bit about these. This is essentially like an evolutionary family tree, is how I kind of tend to think of it as. Here we have 4 different species, and we're trying to depict here their relationships. So we would see all 4 of them share a common ancestor up here that split into 2. One of them gave rise to species 1. The other gave rise to this group that then split and then split again. So you can see 4 and 3 are each other's closest relatives in this, this depiction here. Its closest relative would be these 3, so it shares a common ancestor back there.
So just another question, the trees you're going to be showing us, do we know something about, does Chimera work? Do we know something about the length of these in real time units according to some neutral model of mutation?
So the phylogenies I'm going to be showing are scaled to time, and there's a lot of uncertainty with that. And so a lot of the analyses I did, I tried to account for that uncertainty. So it's going to get a little bit messy, as well as just the relationships themselves. We sometimes struggle with, we might need more and more sequence data to kind of resolve some of these relationships. But yeah, so people eventually, we had an understanding of phylogeny. People started thinking about how some of these symbioses may have evolved. And one of the ideas that came up was that maybe we have, this thing we call co-speciation. The idea being that we could look at a parasite and a host here. And when the host kind of speciates, so does the parasite. Host speciates again, so does the parasite. And so when we compare their evolutionary histories, it's like a mirror. We can see that when one kind of splits, the other kind of follows suit. But we've also recognized that there could be a lot of other situations that go on. There could be extinctions, there could be symbiont switches and things. But the kind of context kind of around the early 2000s was people were kind of hoping there would be good evidence in lichens of this kind of coevolution. Here's kind of a real world example of some gophers and lice. That was kind of a classic example early on where they were seeing, yeah, they're kind of splitting when we get, we get a group of 3 over here, over there, but it's not as clean as people had hoped. And so people finally started looking at lichens in the early 2000s and found, whoa, this is a really kind of a messy affair. We are not seeing any of this kind of pairwise evolution. There's a lot of switching going on here. Yeah, we can see very distantly related fungi on this side, for instance, all kind of partnering with a very closely related group of algae here. It's messy, essentially, was the idea. And there was a series of papers that came out around then. Since then, we've got more and more data. As I mentioned in passing, these algae have been very difficult to identify. And so that's where, like, as sequencing came out, it's really allowed us to use kind of barcoding technologies, et cetera, to reveal much more of this cryptic diversity that's hidden inside those lichens. And so a while back, I had tried to synthesize a bunch of the available data. I focused on this group of algae called Trabuxia. This is the really kind of the popular kid among the lichens. It's the one that is kind of the most common alga in lichen symbioses. And I took kind of all the sequences in GenBank, bin them into OTUs, so kind of species proxies, if you will, on the right here. And I looked at which fungi they were related to and just tried to connect them by lines. And so these lines are connected by the fungal family here on the left. And you can see, same situation, right? It's really, really messy and complicated. We're not seeing a whole lot in the way of this pairwise diversification. But if you kind of squint with me here, everyone, come on. Squint a little bit, you can kind of start seeing some sort of groupings. Like it kind of feels like this group kind of all goes down to this part of the tree. This group kind of, associates with that. You can kind of almost feel like you're starting to see some trends or some patterns there. And so that was kind of the way I'd been thinking about lichen evolution for a long time, was kind of like hoping there'd be this coevolution, this kind of co-diversification. But then kind of said, no, let's kind of step back a little bit here and maybe look at this in from a slightly different perspective. So we go back, to good old Charles Darwin here. Basically, the idea, in origin here is that related things are going to resemble one another. So this is a phylogeny, one of these evolutionary family trees, and I've just kind of used the model here where we have 4 different character states, different colors. And you can see generally that a lot of the relatives here all look alike. They kind of are clustered. So we have those kind of green ones grouping together there, another green clump, another one. So we started out in the blue state. That blue state was retained in many cases to the present here, but we do see some shifts. And once we had those shifts, it oftentimes kind of stays in that new character state. So the green state, et cetera. So yeah, thinking that like, okay, these things should kind of resemble one another. Closely related fungi might kind of resemble one another in this regard. And then I came across some paper by some of Seth Bordenstein's team where they were thinking more about microbiomes, kind of the community of bacteria that are living on or in different organisms. And they were proposing this idea that they were calling phylosymbiosis, basically saying that like the microbial community on some of these animals, if we were kind of to look at the similarity of these communities across different animal species and build kind of a similarity matrix and infer kind of a dendrogram from that, it should kind of reflect the host's phylogeny. And so they cited a number of cases where people were seeing this trend where the microbiome communities kind of recapitulated that host phylogeny. And so here's just kind of the framework they suggested that, if you're working with flies, you could try to create a fly phylogeny here. You could then look at all the microbes living on them, create a distance matrix, looking at, how different the microbial communities are among these different flies. And from that, you could either do some sort of ordination or ultimately make a dendrogram and try to look at, are these communities similar between these relatives there. And so that kind of got me thinking, well, what about lichens? If we start thinking about, the fungus as that animal and these different algae that this fungus partners with as kind of the that microbiome community, do we start seeing anything going on that way? And so a lot of details here that are kind of boring, but ultimately what we did was took a large set of publicly available data of different algal sequences and tried to look at which algae these different fungi were associating with. And then to kind of look at how similar those algal communities were, we used this metric called phylogenetic beta diversity. So we looked not just at the identity of these algae, but how closely related they are to one another. So let's just pretend we have this pretend these are some algae here. We have 3 different species. We have fungus J here and fungus K over here. Both of them are partnering with 2 different algal species. But in this case, this one's partnering with 2 very close relatives, whereas this one is partnering with something that's very different from that other one. And so we can account for that, kind of looking at how unique it is. So we can use kind of some of the branch lengths here to look at how much shared evolutionary history, or how much unique, actually other way around, how much unique evolutionary history do these kind of suites, these pools of algae have from one another. And so we can just kind of do a really simple calculation here where we're looking at the amount of unique diversity for each of these pools. So B plus C here divided by everything there. And we can kind of get this number. So it's a way of kind of calculating this similarity matrix, but accounting for relationships. And what I did then was took this fungal phylogeny and tried to now look at how similar are those algal pools. What does kind of that phylogenetic beta diversity look like? The brighter colors mean they're more similar. Purple means very dissimilar. And so what we wanted to try to see here, just kind of visually, is are closely related fungal species partnering? Are they kind of lighting up yellow? Are they partnering with similar groups of algae? And you can kind of see here, if you look across the diagonal here, you can kind of see some clusters kind of lighting up here.
Wait, sorry, is this algae by algae or algae?
Sorry, this is fungus by fungus. Sorry, so fungus by fungus, so kind of pairwise comparisons. Thank you. Yeah, pairwise comparisons of fungus by fungus. And so what I then tried to do is tried to kind of do a Mantell test. So I wanted to see, as fungal phylogenetic distance increases, try to look at how related are these different fungi to one another, and do these closely related fungi have kind of lower levels of beta diversity, and the others have higher levels. And so for this, I used the number of, this is rather confusing, but there's a lot of uncertainty in our estimation of these phylogenies and the time scales and such. And so I used a number of different approaches. The purple one's kind of the one to focus on. That's the best fungal tree and the best algal tree. And the idea here is that we had a very low correlation when we did one of these Mantell tests, and it wasn't significant. So we're not seeing any sort of significant association between phylogenetic distance and the symbiont dissimilarity. But if we kind of zoom in again, we are seeing some of these clumps here popping up. So I decided to look at a shallower evolutionary scale. So kind of, as Anne had mentioned earlier, if you're looking at different phylogenetic scales, you might get very different patterns or trends emerging. And this is exactly the case here. And so if we look just at one of these fungal families, instead of this very broad range we were looking at previously, we're seeing we do start to see a positive correlation between kind of that distance and dissimilarity. And it is significant. So, okay, that's cool. We're getting there. So what we did then was I decided to break it down into different time classes. Instead of looking at this whole thing, I wanted to do a Mantell correlogram, it's called. The idea being that we're going to break it up into different time bins. And within each time bin, we're going to do a Mantel test here to see, do we see an association between phylogenetic distance and algal community dissimilarity. And the take home here is that at these shallow phylogenetic scales, we were seeing positive correlation there, and it was significant. Same when we did this 25 to 50 million year time bin. But as we went further back in time, that signal started to that association tended to kind of erode. And so it seems like just kind of at shallower scales, we are seeing this kind of hold up where when we look at kind of like a shallow evolutionary time scale, we're seeing that closely related fungi tend to partner with similar suites of algae. But as we go back in time, that starts to erode, and we end up starting to get some convergence even, where we can see very different groups of fungi. So like these and these are very different groups of fungi, but they're partnering with very similar groups of algae. So it kind of starts out, it's kind of I think what's going on is kind of like what Megan was talking about before, where Megan had that cool kind of horseshoe sort of thing going on. And that's something I'm kind of struggling with now, trying to figure out how to sort of deal with. And so, yeah, ultimately what we're kind of seeing here from this is that we are seeing some evidence that these kind of evolutionary legacies are kind of shaping the algal communities that these fungi are partnering with, but that can kind of decay over time. And what I've been trying to do now is trying to add in more variables, because I know there's more than that. I'm trying to add in some climatic and geographic variables, and that's where I'm kind of running into a little bit of trouble as to whether I'm using the appropriate methods or not to try to get at that. But yeah, with that, I just want to thank some of my collaborators who helped, especially with some of the earlier parts of this. And I'm happy to take any questions. Yes.
I have a question about uncertainty. So
yeah,
you're running these statistical tests and they're based on these phylogenies, right? But the phylogenies are based on what we actually observe in terms of species. So in terms of, how should I think about that I have just a partial phylogeny? Is there anything I can do to infer the parts that are missing or something like that for species that I...
Yeah, that's a big problem people have, is that you're absolutely right. This is just the species we've sampled, and there's definitely things that have gone extinct, and we need to kind of integrate those potentially as well. There's things that are still around today that we haven't found as well. And some of it, sometimes it matters, sometimes it doesn't. It somewhat depends on the sorts of analyses people are doing, but that's absolutely a thing and a big issue that arises. And there's a lot of folks, especially at like UChicago, the geology department there, who are really working on trying to integrate extant and extinct data, where they're inferring these phylogenies and they're putting in some of these extinct species as well, because when they try to model trait evolution, they've shown that this really can have a substantial impact. At this point, I'm not entirely clear how to add them in. I think you could perhaps do some sort of approach where you are kind of you could use some of like the methods Anchow's developed, essentially, to try to figure out like what the total standing diversity might be and try to plop that in. But for now, when I was using that uncertainty, it was mainly uncertainty in the ages of these different groups as well as the relationships. And so trying to kind of be cognizant of that. Yes.
Do the fungi and algae species that exist in lichens only exist in lichens, or are the ones that also exist outside?
The fungi and algae that are in lichens only exist in lichens, or can they go it on their own? The algae, I would say yes. That's been kind of controversial. There's been some hardliners in the past who said no, they're so adapted to living in this buffered situation, they can't live on their own. And then people would find it, and he'd say, well, that's just escaped. That's not going to survive over the long term. And people kept finding it and finding it. And I think they they have to be out there. More and more people are finding them. The fungi, on the other hand, is where it gets much murkier. There's a handful that do live on their own that we have good evidence that they do live on their own. The rest of them, so there's a couple of ways some of these lichens will disperse. And one of them, you know how Anne was talking yesterday about mushrooms, and she was saying that some of these other fungi make those like sacs, those asci that contain 8 spores. Lichen fungi tend to be in that group with those 8 spores. And so they will shoot their spores off independently, and they have to acquire a new algal symbiont. And that's sort of one of the questions is, okay, when that spore germinates, how long can it persist without its algal symbiont? And it's kind of a black box for us, unfortunately. And this is one of those cases where experiments would be awesome, but they're really tricky. There's some suggestions that maybe they could partner with an algal symbiont that is not the ideal partner and kind of parasitize it for a while. And then hopefully a compatible partner blows along. But it's really unclear to us. And that's kind of coupled with, I don't know if you remember in the beginning, I showed like a picture of what that fungus looked like when it's on its own and kind of made a reference to Jabba the Hutt. It's like, would we even recognize it in nature if we saw that? That's kind of the other thing. But at the same time, people have started sequencing the genomes of these fungi and are finding pretty much exactly what they were finding with some of these ectomycorrhizal fungi. So those are fungi that partner with tree roots. And so when people are looking at the genomes of these lichen fungi, they were finding, holy crap, there's a lot of genes responsible for breaking down plant cell walls in here, more than we would kind of expect. If these lichens had shifted to strictly depending on these algae and having those algae leak all of that, they shouldn't still have genes there that help them break down lignin or cellulose and things like that. That should kind of all get kind of weeded out over 100 million years or 200 million years. But we end up seeing there's a lot of them still left, which is what they were seeing kind of in these ectomycorrhizal fungi. And they're like, maybe they're possibly getting carbon somewhere else, not just from their hosts. So it's raising this question now of, are these lichen fungi, do they have the genes that would allow them to kind of get food from a log in the interim, perhaps, and maybe survive a while? So it's really kind of a black box at this point. But really, as I'm guessing it always is in science, as these new tools are coming out, we just keep having, being able to examine some of these longstanding questions with new techniques. And yeah, it's been exciting to see what people keep finding. Do we have time for one more?
That's your question. We'll get Ben set up.
Sounds good. Yes.
So, you briefly mentioned does it matter of the substrates? Like, you talk about symbiosis between fungi and algae, but you briefly mentioned the substrate a little bit, but yeah, the only angle on the substrate is also important. So, is there any the evolution it's not symbiosis, but it's a more like a substrate dependent kind of
There's absolutely some of that. So individual lichens tend to have preferences. So some will prefer to grow on tree bark, some want smooth tree bark, some want like on a twig, some want to be in high sunlight on the canopies, some want to be in shadier spots, some on rocks, some even on leaves. So there is some kind of specificity there in terms of the substrate. And there's some generalists as well. But, we have that, and that's in terms of the kind of relichenization step. That's something that's some folks have raised kind of question about is we need to think about these algae too. If they are living in the free living state and now will get acquired by this fungus, are they able to tolerate that acidic rock or not? And they're going to have these preferences. Well, like you're getting at. I don't know if that can answer that.
Yeah, it's
approximated.
Thanks, Matt.
Thank you, Matt.